Good evening, ladies and gentlemen. Welcome to this very special occasion, this public lecture hosted by the Sleep and Circadian Neuroscience Institute, and where we will, after Professor Pack's lecture, be very proudly presenting Allan with a medal to commemorate his lifetime achievement and contribution to sleep and circadian science. And Allan's going to be talking to us on sleep loss and sleep disorders. I'm Colin Espy. I'm putting on a special Scottish accent this evening, particularly for Allan, who is of Scottish stock. And this is Allan, I guess, a few years ago. I mean, I've known Allan for a long time. I have to say, not as long as this, Allan. This goes back, I guess, to medical school days. And this is, as many people might recognise, a scene from Scotland. This is, in fact, Loch Lomond. And Allan grew up in an area called the Vale of Leven, or as we call it affectionately locally, the Vale, which seems sufficient sometimes to describe the area. But its hinterland is absolutely beautiful. And Allan spent a bit of his early life on this boat, which is a paddle steamer called the Maid of the Loch, and I believe you were a purser on this. I guess you earned at least a couple of pounds a month or something doing that in your student days. But I think there was interest also in another maid of the Loch. We're not talking about Allan's wife, Fran, who's apparently heard Allan speak often enough, so she's having a refreshment and will join us later on. But it's wonderful to have Allan and Fran here to celebrate this special occasion. I'm also very proud that Allan went to the University of Glasgow. You know, I don't think he was in on the Sgt. Pepper album, but I think the album came out the same year as you graduated in medicine, Allan, from the University of Glasgow, and went on afterwards to do your PhD in mathematical models of lung function. And Glasgow, Allan, is very proud of you, as is Oxford. One of the things that every time I see Allan, and I've been fortunate to be in Allan's company on other occasions and to have hospitality in your home in Philadelphia, that Allan runs his operation, which is very considerable, sizable, and very effective, just like the family. There is the family. There's Allan as a younger person, but there is a small part of the academic family. And everyone who knows Allan knows his generosity in giving time, encouragement, mentorship to so many people. And the legacy of great people is often the people they've influenced. And Allan, we're very grateful for all the scientific work you've done, and indeed for the people you've influenced over your career. This is Allan with a touch of tartan when he spoke in Glasgow I think in 2008 at the European Sleep Research Society meeting. And Allan, it's wonderful to have you here tonight to speak to us on sleep loss and sleep disorders, a major unmet public health need.
Thank you very much for that very generous introduction. I mean, actually, that boat there, the Maid of the Lark, I met with my wife in that boat. She took a day trip, and she got more than she bargained for. Anyway, so let me just get going here. So what I'm going to talk about here, I'm going to talk about what is circadian rhythm, what is sleep, why do we sleep, what happens when we're sleep deprived. I'll talk a little about common sleep disorders, and then why companies are starting to really care about sleep in their employees. The basic idea of sleep is shown here, that you have this two-process model. You have the circadian thing, the C, which oscillates with a rhythm that's slightly different than 24 hours. And then you have this process S, which is the sleep drive system. And the way we think about process S is that the drive for sleep is very low after you've slept, and it progressively increases the longer you're awake. For a while, it's opposed by an alertness signal that comes from the clock. And then late at night, the alertness signal drops off. You've got a high sleep drive. You go to sleep, and then, as you can see, process S is restored. So it's like an old-fashioned egg timer. The sands are building up. That's the drive for sleep. And then when you go to sleep, you flip it, and you recover, and you're back to deal with a new day. Every cell, actually, one of the amazing things that's been shown over the last several years is that every cell in the body actually has a clock. There's an intrinsic clock in every individual cell. And what happens is that clock is controlling the expression of many, many genes. In addition, what happens is sleep-wake system is also coming in with signals to that local clock and is influencing in some way, we don't know how, but it's clear it does so, and also patterns of food intake. So sleep-wake, food intake, and then these local clock. But the important thing is there's a diurnal rhythm to expression of many, many genes. Some would argue up to 50%, depends how you define diurnal, but certainly a conservative estimate would be about 15%. And so what that means is that your organs, your heart, your lung, your liver, and so on, they're changing across the day. They're doing different things, and there's this temporal control. And one of the great things about the circadian world is thinking about the implications of that temporal control. So the functions of every organ varies across the day. But there is actually a master clock. So here's the master clock, and it's in the suprachiasmatic nucleus. He's the conductor of this clock orchestra, if you like. And what we know about the master clock is it's situated in the suprachiasmatic nucleus of the hypothalamus here. And then the major entrainer to the clock is the light-dark cycle, to the master clock. There's light darks. The intrinsic rhythm of the clock is slightly more than 24 hours, and then the light dark cycle pulls it into 24 hours. And then if you change time zones and you're out of sync, it's the light dark cycle that's re-entraining you. And basically, one of the major accomplishments of modern sort of molecular biology and behavioral work was the description of how the clock actually ticks. So we know that the clock ticks in a molecular way, and I'll explain a bit more about that in a minute. So there's a light dark, a path from the eye to the brain into the suprachiasmatic nucleus. Now, the light sensing, as I've said, entrains the clock. One of the great discoveries from the Oxford group was the fact that there's an entirely separate light sensing mechanism for the clock compared to vision. Dr. Forster's group created a mouse with knocked out a particular transcription factor. It didn't have rods and cones, so it couldn't see. It was visually blind. But the amazing thing was, it must have been quite an interesting experience to see that mouse entrained perfectly normally to light and dark. So the light sensing for the clock was clearly different. Russell went on to identify it was in these retinal ganglion cells which are across the retina. It's a particular pigment called melanopsin, and that's what's sensed, and that drives the retinal hypothalamic tract. There's a tract right from the retina to the suprachiasmatic nucleus. One of the things about melanopsin is that melanopsin particularly senses blue light. It's very sensitive to blue light. And although Dr. Forster didn't intend to do this, he actually created a whole cottage industry of blue light. You can get blue light glasses, you can get blue light blocking glasses. So if you want to not get light in to keep you awake, you can wear these blue light blocking glasses. Last year, we were asked by the Eagles, which is a local NFL American football team, to come and give them lectures about sleep, because the coach in particular was very, very committed to the idea that good sleep was essential for performance, athletic performance. And he'd read so much about this that he had every Eagles player sleeping with the blue light blocking glasses on the night before the game. They didn't do too well because it didn't make it. And in the local sports shows, they kept saying he should stop worrying about the sleep and start worrying about what they're doing in the field. They might get on better. But now, in fact, some of these the computers and so on actually at a certain time of day filter out the blue light. So that you're not kept alert after midnight with blue light coming from your computer. So it's really, and we can manipulate the clock pretty well with this, with blue light and blue light blocking glasses, and there's lots of opportunities. So circadian rhythm, as I've indicated, genes oscillate over 24 hours, the circadian period, and we know there's clock proteins, and the clock proteins feed back into the nucleus to inhibit their own transcription, so they turn themselves off. So the mechanism is at core, it's a negative feedback mechanism. There's other mechanisms, other loops and negative and positive involved, but the core of it is this negative feedback involved, in particular clock genes and clock proteins. Sleep, the concept about sleep is that there is an accumulation. This is the idea. Some people would challenge this. There's an accumulation of sleep-promoting molecules that drive sleep. So they're building up during the day, and then when the clock goes down, then you get to sleep. One of the sleep-promoting molecules is adenosine. I mean, that's been known for a while. And one of the reasons we drink all this coffee is it antagonizes the action of adenosine. You could actually argue that Starbucks is the biggest pharmaceutical chain on the planet. It's selling this profound weight-promoting molecule, and there's probably other molecules. Understanding the basis of sleep drive, sleep homeostasis is called, is one of the goals of modern biology in this field, because the clock has been so successful. One of the key things is that different animals, every animal has a clock close to 24 hours, but different animals are programmed to stay awake for different times. A mouse, the most you'll ever see a mouse staying awake is about 3 hours, and then it falls asleep. Whereas humans are actually programmed to stay awake for about 16 hours. So we can stay awake for 16 hours, fine. Once you get beyond that, you start getting impaired. And the further you're beyond that, the more impaired you actually become. And we believe that sleep actually helps in the consolidation of memory. Now, one of the things that's happened over the last, maybe 10, 15 years is we've begun to identify sleep in non-mammalian systems. Most of sleep research up to the year 2000 was in mammals, mouse, rats, and originally in cats. And then our group, and also Paul Shaw group, recognized that there was sleep in Drosophila and fruit flies. We come up with different definitions. We weren't doing EEGs in Drosophila, behavioral definitions. And since then, sleep has now been identified in zebrafish, and also David Raisin at Penn identified sleep in C. elegans, the worm. So you've got every single model system out there. And what's happening is you're beginning to see that the pathways that are changing with sleep are changing across all these different systems, showing the conservation not only of the state, but we believe of the molecular mechanism. One of the reasons for studying these simple animals is you can do the experiments much faster, and you can get answers much quicker. In Drosophila, there's wonderful molecular genetic tools that allow you to screen and find genes. In C. elegans, there's only 302 neurons. And every neuron is known, and the connection of every neuron is known. And we know there's one neuron, the ALA neuron, that plays a particular role in driving sleep. So it's got huge advantages in terms of a simple circuit. The great advantage in zebrafish is the transparency. So we've got all these different systems now to study sleep. In mammalian systems, and mammals, what we know is it's not just a homogeneous state. There's actually different states built in within it. Nobody, for the moment, has really described convincingly that there's different states in Drosophila and zebrafish, and we're not really sure. But certainly in mammalian systems, there are these different states. Whether they evolved later, who knows? But basically, we can talk about states in terms of non-REM and REM sleep, R for REM sleep. And this cycle is very different in mice. The mouse cycle very rapidly in a few minutes. The human cycle is about 90 minutes. So you get into REM, 90 minutes, another episode of REM 90 minutes later. And as the night goes on, the episodes of REM get longer. And they also get more phasic. You know, the eyes are moving more rapidly. So basically what happens is you have non-REM sleep, and it's divided into 3 stages: light sleep, transition sleep, and then the deep sleep, these slow waves, big slow waves you see in the EEG. And then you get rapid eye movement sleep, REM sleep. This is a stage of sleep that we believe you dream, because you wake people up and they recall their dreams. But you're actively paralyzed in REM sleep, so you don't live out your dreams. Your brain's having a great time, but you don't actually move. There are people, there's a so-called REM behavior disorder, where people lose the atonia, the paralysis of REM sleep, and they can act out their dreams, often in a quite vicious way. So there's these sub-stages of sleep with a wonderful organized rhythm. Now, there's a very important interaction between the sleep-wake and the circadian system that controls the rhythm of sleepiness across the day. When you wake up in the morning, point 1 here, you're wide alert, you've slept, you've recovered that whole thing, the process S is at a very low level, and you're good to go. But then what happens is, right after lunch, you actually get sleepy again. That's the siesta time. So you see this siesta occurs after lunch. In Spanish cultures, they obviously take naps and they sleep. In American cultures, we don't do that, and probably in the UK, you don't do that. You just push through that. And even though you haven't slept, you become alert again because the rhythm is coming back. Interestingly, that siesta rhythm is also seen in Drosophila. You see exactly the same thing. You see sleep during the middle of the day, you see sleep at night, and so on. So that rhythm, that double period of sleepiness, if you like, is conserved in Drosophila as well. And then what happens, as I said, even though you haven't slept, in the early evening, it's often called the forbidden zone for sleep, because you're really pushed to be alert at that point, and it's just hard to sleep. Then as you go on, when you get to the later in the night, the clock goes down, you become sleepy again, and you go to sleep. So that's how it works. And really, one of the important things is you can't just sleep at will. There's this myth out there that you can just sleep when you want. The clock is really setting the timing window in which you can sleep. And if you're trying to sleep at times at which the clock is telling you to be alert, you actually have impaired sleep efficiency. It's really difficult to sleep, and you don't sleep particularly well. Now, the timing of sleep varies between people. Right? So we talked about how the clock sets the timing, but there are differences between us in terms of this timing. And we know this is genetic. Recently, there's been some variants, not surprisingly, of clock genes recognized that control this. So some people are larks. These are people who go early to bed. They're not party people. These are not, and they get up early in the morning and have a biological advantage. Almost every single dean we've had at Penn in the last few years has been a lark. And Bill Kelly, who's the transformational dean at Penn, when he came, he started organizing 6:30 a.m. meetings. And the chairman of radiology, when he was asked to go to a 6:30 a.m. meeting, said, "No, I'm sorry, my schedule's full at that time, but I can come at 5:30. So anyway, so there's larks, and then there's people like me, we're owls, we stay up late. 7 a.m. meeting kill me. I get in there for that. It screws my whole day. So there are these differences, and these are the extremes. And then there's most people are in the middle. I always think it's very important for people when they're dating their spouses or future spouses, they should sort of line up and make sure the time is fairly close, because you may not see too much of your mate if you're both at the extreme. And it's hard to sleep if you sleep at the wrong circadian time. The implications of that are both for shift work, and increasingly the corporate world is recognizing the implications for jet lag. What happens to night shift workers is the following. When they go home in the morning, right, they're very, very sleepy because they've been up all night, and they get to sleep fairly quickly, but they can't sustain sleep because the clock is saying, no, you're supposed to be awake. And so they wake up about 1 o'clock, somewhere around about there. And then they can't get back to sleep. And then they're awake for a very long time till the next morning. And so the difficult thing for shift workers, and it's really dangerous, is the drive home first thing in the morning, because they're very, very sleepy at that point. And, a number of them crash and so on. Some places now are using taxis. They're not letting them drive home. They drive them home to avoid that risk. Many years ago, I was in a task force in New York about drowsy driving. And we were talking to him about educational programs in New York State. And I said, well, one of the big risk groups is shift workers. So they did this focus group of shift workers. They brought 20 shift workers in just to hear their stories and so on. And what happened was every one of them talked about how difficult it was driving home in the morning. And there was one guy there, he said, what he did is he said he took a bag of ice with him. And then as he was driving home and feeling sleepy, he was applying this ice to various parts of his anatomy just to just to keep himself awake. So he had his own countermeasure. I'm not saying we should be widely proposing that. And then jet lag. And jet lag, if you go to Japan and you're 12 hours out of sync, I mean, the clock is completely out of sync, and it's very difficult to sleep. And then it adjusts very slowly. It only adjusts about 1 hour a day, so it takes a while to actually readjust. It's the light dark cycle that's readjusting you. Now, that's the rhythm I talked about, nighttime, middle of the day, and so on. We published this paper years ago about the time of car crashes from falling asleep, and just to look at what that looked like. Was there a specific rhythm to that? We did this in the state of North Carolina, and we did it in North Carolina for two reasons. One reason was they had a uniform crash reporting system across the whole state, and secondly, they had a box in the form where the policeman could tick fell asleep. Now, we don't know how he made that judgment, and there was no alcohol involved. But what you can see here is this lovely rhythm, and this is in people between 16 and 25, right? So this is midnight. You can see the number of crashes going up and up and up. You get in the morning, and then it drops off dramatically. You think there may be a little blip in the afternoon. You can see the forbidden zone here. There's almost no fall asleep crashes, and then it picks up again. So the rhythm I explained is actually seen, as you can see here, in real life. When you looked at crashes, time of crashes, falling asleep of people over the age of 65, it was a different rhythm. Here it's shown here. And you can see there was nothing at the night time. The reason for that is when I was a young person, I'd be driving around at 2 in the morning maybe, and there's never doing that anymore. So you get to a certain age and you just don't drive at night. And so what you can see is all that's left, and the numbers are not as big of crashes, but all that's left is the siesta time. So you can see the siesta actually coming through. The point of showing that is that while you think about this theoretical thing about these rhythms of sleepiness, they really are reflected in real life world circumstances. Now, why do we sleep? This is one of the big questions of modern sleep biology. We spend a third of our lives doing it. We do a lot of this, right? And Rex Stafford once said, he was one of our early investigators, if there's no reason for sleep, sleep is the largest evolutionary mistake that was ever made. If we're spending a third of our life doing it, and it's not doing any good for us, maybe this is not a good thing. And so if there's a reason to sleep, most people, including ourselves, what we've looked at genes changing expression between sleep and wake, trying to get clues as to what the functions of sleep are. And I think there's certainly a number of very viable hypotheses now based on molecular studies and others about the functions of sleep. One of the theories that's gained a lot of attraction is the so-called synaptic homeostasis theory. Because what you find is that genes that are involved in scaling up synapses like BNP, ARC, and so on, they upregulate during wakefulness and they go down during sleep. And that led to the idea, this is Tononi's and Sorelli's work, that basically what happens is during the day, you're making connections, things are happening, you're making connections, you're strengthening connections, and then at night you prune the connections. You don't keep making connections for the rest of your life. You have to prune them, but you're still leaving a trace, essentially, from what happened to you during the day. So that's one of the major theories, the synaptic homeostasis theory, and it came, there's evidence to support it in different ways, but it came originally from studies in gene expression in rats. A paper we published showed macromolecule biosynthesis. What again we looked at was genes that were increased expression during sleep compared to wakefulness when the animal was sacrificed at the same time of day. We deprived them, made sure they were lined up at exactly the same time of day. And what we found was multiple genes involved in protein synthesis, lipid synthesis for membranes were upregulated during sleep. It raises the interesting idea that what sleep is about is basically a metabolic cycle. That what you're doing during the day, the energy you have available, you're using it to man the pumps and your neurons are firing. And then when you go to sleep, you take that energy and you switch it and you start making things to be ready for the new day. So basically, it's a restorative stage, as you can imagine. Another concept that's out there, we've certainly played a role in this, Jeannie do in particular, is that when you get sleep deprived, sleep deprivation is not innocuous. What you get is what's called the unfolded protein response. All the genes involved in that whole pathway is upregulated if you sleep deprived mice for about 6 hours. So basically what this is the cell, if you're making proteins, you need to fold them properly. But then if the cell is under stress, then it can't fold these proteins properly and they start misfolding. If they misfold, they can form aggregates and be very toxic to the cell. So the cell has a mechanism to deal with that. The so-called unfolded protein response. It's basically a defense mechanism. It says, oh, we're misfolding proteins here. It then changes BIP, for example, which helps proteins get folded, and it tries to shut down protein translation. So it tries to stop making more proteins, because if they're misfolding, you don't want to make more of them. That whole pathway is activated in mouse brain with about 6 hours of sleep deprivation. So it really indicates that when you keep awake for longer than is normal, it's not just your performance is changing, you get molecular consequences. And we're beginning to think that sleep deprivation is not that innocuous from this point of view. But sleep only for the brain. There's a famous paper from Allan I, which was in Nature, which was entitled, "Sleep is by the brain and for the brain." Is that true? Is sleep only for the brain? Are we only sleeping for the brain? So we did recently a study, again, we're looking at genes changing between heart and lung now, between sleep and wakefulness. And what we then did is we deprived animals, we allowed them to sleep, and we sacrificed them at exactly the same time of day. And then we said, what genes are changing? And what we found was the overrepresented genes were all these protein folding genes. It was very similar to brain. You can see the chaperones, which help proteins to fold, the protein folding, the stress response, that this is the degree of enrichment of these, and that's the p-value. So they were all changing, and they were changing in a very similar way, both in heart and lung, and it was very similar to brain. So there has to be some mechanism that's associated with sleep that's coordinating that out there in the periphery. So we don't believe that sleep is just for the brain. We believe it has a function also for peripheral organs. Another concept that comes out of Neerdegaard's work is the idea that sleep is also about removing toxins. I talked about how one idea is that sleep is restoring things, it's making new proteins, it's making new lipids and membranes and so on. This idea is that the brain is actually clearing things during sleep. And this is a kind of a seminal paper that was published in Science. And the green actually is a sleeping brain, and the red is an awake brain. And what this is showing is that if you put compounds in there, label compounds, and you see how much they move, during wakefulness, they hardly move at all. They just stick there. What happens during sleep is a huge convective movement across the brain. And it's thought that what there is upregulation channels like aquaporin, they all connect, and the whole brain then becomes this pumping system that can really take things out. And it actually clears molecules. So that's one of the ideas. You can see the clearance is shown here. The orange line at the top, that's during wakefulness. The sleep is the green, and this is anesthetics, which are very similar. And what they're looking at is they've injected into the brain beta-amyloid, which builds up in Alzheimer's. It's a labeled beta-amyloid. And you look at the clearance of this over time. So there it's cleared during wakefulness. You kept the mouse awake. Here it is going down, and it clears much more rapidly when you're asleep. So that's the idea then, is that the sleep is actually helping take things, toxic things out of the brain. It's building new things for the next day, taking things out of the day, out of the brain. And so the trash trucks for the brain, the notion is they arrive during sleep. They're the guys, getting the stuff out of there. So these are the theories that are out there. There's a number of theories. There's increasing data to support them. Some people believe there's going to be one core function for sleep. I don't believe that. I believe there's going to be a number of functions for sleep, and it's going to be interesting to show that these functions we're talking about are really replicated in the lower species. Every single animal, whether it's been birds, Drosophila, mice, rats that have been sleep deprived, they all show the unfolded protein response. That is a ubiquitous finding with sleep deprivation. Now, there are major types of sleep loss. So the first type of sleep loss would be short term. What I said is that you can stay awake for 16 hours. If you try to stay awake longer than that, you'll get impaired, and the longer you try to stay awake, the more impaired you'll become. Long term, in chronic insufficient sleep, that's an extremely common phenomenon. So about a third of the population in the US. And what that means is that you get inadequate sleep day after day. What often happens is it involves catch-up sleep at weekends. So you get up early during the week, you really cut back in your sleep, and then you sleep in longer at the weekend, and you do this catch-up. The CDC in the States is now monitoring admittedly, self-reported sleep duration in different states. And they've got maps now, just like the maps of obesity rates, where the most obese states are. They've also got maps of short sleep, and they're quite interesting. They sort of line up over each other. The more obese states are also got ones with the shorter sleep duration. But obviously, both short and long-term sleep loss, you may have chronic sleep loss and then have a period of a really, long period where you don't sleep, so they can coexist. Now, this slide, it comes from David Dinge's group at Penn. It's a fairly similar slide, this. And what David did was he looked to ask the question, what was the response to sleep deprivation, sleep loss? Was that different between people? And was it a biological trait? And what's plotted here is on the y-axis is PVT lapses. So this is PVT lapses over here. It's a test that David developed. The idea is, and I'll mention a bit more about this in a minute, that when you're really sleepy, wake state is unstable. You just can't sustain wakefulness. And you've been there. You get these microsleeps, you drift in and out of sleep. And you can pick up this with this test, a psychomotor vigilance reaction time test, where basically a box is there and a light comes on at random intervals and you respond and you measure the reaction. Now, if you're drifting to sleep, you don't respond. And a lapse is called a reaction time of over 500 milliseconds, and you count that as a lapse. And this was done every 2 hours across a 30-hour period of sleep deprivation, so that would be 19 trials, and you can see the total number of lapses. Now, what's plotted in the bottom here are the different subjects, and the rank order from the person with the lowest lapses to the person with the most lapses, okay? And here. And what he did, he sent them home, and he repeated the whole thing a few weeks later. And what you can see is the two trials were right on top of each other, right? So this person here had very few, the end there had very few lapses in the red star, one trial, and then almost none in the blue trial. Whereas the person at the other end, K, had huge number of lapses in both trials. So it was clear it was very much a biological trait. And the interclass correlation coefficient was like 0.8. We then did a twin study where we did exactly the same thing here, and we showed that it was an extremely heritable trait. The heritability was about 80%. So just as there are genes that control when you go to sleep, there are genes that control how you're affected by sleep deprivation. Some people are very resistant, some are very sensitive. In fact, we know that there's a gene involved in this. These are not huge numbers of subjects, but it's a proof of concept. It's a clock-associated gene called DEC2. And the original mutation was described in the He paper in Science in 2009, and then we described another mutation in the same exon in 2014. And what these mutations in that gene lead to is they lead to short sleep. They sleep less than 6 hours, but they've got no daytime impairment. So they're the classic sort of short sleepers. They have no daytime impairment. They have an increased resistance to sleep deprivation. In our case, we were looking at the twins, and we had the dizygotic pair, one who had the mutation, one who didn't. The one who had the mutation slept short, but when we sleep deprived them, had a lot less lapses. So it wasn't, they were sleeping short and they were resistant to the effects of sleep deprivation. That twin also had decreased REM sleep. The amount of non-REM sleep was relatively maintained. And the beautiful part about the Science paper was they took that mutation, the human mutation, and they knocked it into a mouse, and the mouse slept short. And then when you deprived the mouse, it had less recovery sleep. So you could actually replicate the human phenotype in the mouse model with that mutation. So this is definitely a mutation that leads to short sleep. Now, different people have called it different things, right? So when we published our paper, the BBC called it the Thatcher gene, because when Margaret Thatcher was in office, she was only supposed to sleep 3 or 4 hours a night. And then recently I got an email from a political reporter in Politico who said to me, do you think that Donald Trump might have this gene because he doesn't sleep very, very long at night either? I said, well, you just have to think about it. I mean, certainly that's possible, but the more common explanation of people who sleep short is they're not sleeping enough and they're cognitively impaired. So you just have to decide which way you want to go there, right? So anyway. Now, sleep and performance, I talked about acute sleep loss. Let's talk about this chronic sleep thing. And this is a paper from Hans van Dongen. And what they did is they brought people into a lab and they gave them a certain amount of sleep a night, 8 hours, 6 hours, or 4 hours. And so they were getting progressively sleep deprived. And again, what we're measuring here are these performance lapses. So what you can see is that, the less sleep you get, the more impaired. But what you also see is cumulative. It never stops. It just keeps getting worse. I mean, people thought, well, a couple of nights of that, you'll adapt. You don't. The performance just keeps getting worse and worse over these 14 days. So after two weeks, you don't adjust, you just get worse. So it's a cumulative phenomenon, and that's the way we think about it. People the way you behave today isn't just dependent on how you slept last night, but the previous couple of weeks of sleep. If you haven't slept in the last couple of weeks if you slept short, you're going to be having these performance lapses. What's interesting is there's the objective data on one side, and the subject, how sleepy they feel. And what you can see is, if you look at the 4 hours and the 6 hours, the performance is just keeping getting worse and worse. But if you look at the subjective, it goes up for a couple of days and then flattens off. And so the gap between how sleepy you think you are and how sleepy you actually are gets bigger and bigger. So you can't rely on self-perception of sleepiness to know how sleepy you are. You can see there's a real difference in the shape of these curves. Sleep deprivation affects your ability to stay awake. So wake state is unstable, and I've mentioned that. Because what you get is you just drift off to sleep. You get these involuntary microsleeps occur. Now, if you're sitting in this audience and you have a microsleep, it's not a big deal. You miss a slide or something, and you come to for the next talk, the next slide. But if you're driving your car, in that microsleep, you can be right off the road into a bridge abutment and do serious damage to yourself. So the consequences of involuntary microsleeps really depend on circumstance. Attention-intensive performance is unstable, so if you really have to be attending to the task, it's hard because you're drifting. You have increased errors, lapses, and you keep trying to compensate for that, and you can have wrong responses. So as a fundamental concept, the wake state is unstable if you're sleep deprived. You also get cognitive slowing. You just don't respond as quickly. Your response time slows. It takes longer to complete tasks. If you have to complete it in a certain time, you have much more errors. But it's not just behavior. Sleep deprivation also affects your heart and metabolism. It's chronic sleep deprivation, shift work, and so on is a risk factor for hypertension, vascular disease. We know we can do this in people like you. I could bring you into a lab. I could give you 4 hours of sleep for a week. And what I would do at the end of that week, if I did a glucose tolerance test, you would be like a pre-diabetic. You develop insulin resistance. And you can see that in a simulated way in labs. And we also know that what happens is your leptin levels get depressed. That's the thing that turns off your appetite. Ghrelin, which turns on your appetite, goes up. And so you actually eat more, and you tend to eat higher calorie foods and so on. And that's one of the reasons we believe that chronic sleep loss is a risk factor for obesity. And there's certainly data out there to show that if you look at the people sleeping 5 hours, they've got a higher rate of obesity than people sleeping 7 to 8 hours. There also are common sleep disorders. A lot of this we're just talking about in terms of sleep loss is people choosing not to sleep and doing other things. Now we're talking about common sleep disorders. The first is chronic insufficient sleep, we just talked about. There's a common sleep disorder called obstructive sleep apnea. There's another one called insomnia, where you have difficulty sleeping. Everybody has transient insomnia. You sleep in a different hotel one night, you don't get to sleep so well. We're not talking about that. We're talking about people who have that on a chronic basis, night after night for months. Shift work sleep disorder, we mentioned that. People who are doing shift work, they can be very sleepy. And there's a syndrome called restless legs syndrome, where you get these weird sensations in your legs just before you go to sleep, and it can make it difficult to sleep. And these are all extremely common problems, and they're all related to specific biology. The disease I study the most, or our group studies the most, is sleep apnea. And let me just explain what that is and some of the information about that. So what you can see here in this side, you can see an awake airway. This is a sagittal section. Here's your tongue, and here's your airway. It's open. Now you go to sleep, and what happens is, just like you lose the tone in your arms and legs when you go to sleep, you lose the muscle tone in your upper airway. And when you lose the muscle tone in your upper airway, the airway narrows. That happens in everybody. But in some people, it narrows so much it actually blocks off, right? And so what happens is they fall asleep, their airway blocks, they're continuing to try to breathe, their diaphragm is really working. What's happening, they're not breathing, the oxygen level's declining, the CO2 level is rising, and the brain senses that and wakes you to a lighter stage of sleep. And when you wake up to that lighter stage of sleep, it sends a big signal out to these airway dilator muscles, and the airway opens up. So, and now what happens, you go back to sleep again, and you go this way again. So you just keep flipping back and forward, and you have these apneas at times when there's absolutely no breathing at all. And it's an obstruction at the back of the tongue there is what happens. Is sleep apnea treatable? The answer is yes, it's treatable. It's a very crude treatment, but incredibly effective. And the instrumentation for this has improved over the years. The basic idea is, Colin Sullivan in Australia developed this in 1981. The basic idea is you just wear a mask over your nose, right? And you apply air under pressure, right? So here's the airway blocked. I put this mask on, I've got a certain amount of pressure, not oxygen, just air under pressure. I pressurize the back of your throat there, pushes the airway open, you're good. The snoring disappears, the apneas disappear, and you're good to go. So this is a mega industry now worldwide because of the number of people with sleep apnea. It's a very simple treatment and extremely effective with almost no risk, almost no side effects. What happens when you have sleep apnea is the following. So the green here in this part, sorry, to this side is somebody who's sleeping without CPAP, and this is somebody with CPAP over here. Now what happens is, this is the hypnogram at the top with different stages of sleep, and you can see the spikes. So they're changing state frequently. So their sleep is extremely interrupted. It's there's no continuous sleep. It's very, very interrupted. Then what happens here is we put them on CPAP, we keep their airway open, and you can see the difference. Now, what you can see here is they'll suddenly get these episodes of REM sleep. And these people haven't been in REM sleep for years, because every time they tried to get into REM sleep, they would get an apnea, it would kick them out. And now they've got this incredible pressure for REM sleep. And you can see they have these huge long episodes of rapid eye movement sleep when they're on CPAP. And then when these people wake up in the morning, it's like a religious experience. They say, what happened? I haven't felt like this in years. I mean, they're just it's so dramatic, actually. And then here, you can see the oxygen saturations here. So the oxygen there, it drops, it goes up, and it cycles. And as it cycles, with this cycling, it produces a lot of free radicals, and that's what's doing the damage to the thing. You put them in CPAP, and it's perfectly stable. So you can really turn this thing around quite dramatically. The reason Scalia is pictured is here is Justice Scalia, died during his sleep in the last year. It caused a huge political storm in the United States because he hasn't been replaced in the Supreme Court. Not surprisingly, when you see him, you would say, well, maybe he did have sleep apnea. Actually, he died in his bed, and the Washington Post reported there was a CPAP machine on his bedside table, but he wasn't wearing it. So when he died that night, he obviously had severe sleep apnea, was not wearing his CPAP. Now, whether that contributed to his death, who knows, but it does seem extremely likely. Now, sleep apnea is a risk factor because that oxygen, that dropping and cycling of oxygen is seen by every single tissue of the body. Sleep apnea is a risk factor for a lot of things. It's a risk factor, not surprisingly, for car crashes. You're really sleepy. Even though you've slept 8 hours, you don't get restored to sleep. You're incredibly sleepy. You fall asleep driving. It's also a risk factor for a number of cardiovascular disease, for diabetes. And there's recent data showing that if you have Alzheimer's with sleep apnea, it just moves much more rapidly. The process of neurodegeneration proceeds much more rapidly. So it's got a lot of systemic effects, essentially. Now, we know that the major risk factor for sleep apnea is obesity, and you can see the obesity rates here increasing. And this is the prevalence data for sleep apnea. The classic paper was published in Wisconsin, 1994. 4% of men, 2% of women. Now you can see we're talking somewhere around 10, 17%, depending on the age. So the prevalence of sleep apnea has gone up and up and up. This is a very, very common disorder. Now, what's the link? Why is there a link between obesity and sleep apnea? And I mean, it sounds weird talking about this, but could it be fat deposited in the tongue? And that's one of the things we think about, that this is the connection. The reason we think about this came from this picture here. I collaborate with people at Jackson Labs up in Maine, the mouse genetics place, and they showed me this picture of a New Zealand obese mouse. And it's a polygenetic abnormality in a mouse. It's the fattest mouse that's out there. And they showed me this picture. This mouse sleeps standing up, right? So he puts his paw and he sleeps standing up. And I said, well, he's obviously got sleep apnea, his airway, and indeed that's what he's got. He's really, that mouse has sleep apnea. And he's got fat deposited right in his tongue. He's got a much larger tongue and a much smaller airway. So that's what we think the link is. And then Rich Schwab at Penn recently has developed an imaging strategy where we can actually image people and we can calculate the amount of fat in your tongue, which is the yellow here. And this is somebody who's got these two people with the same BMI. This person's got severe sleep apnea, this person doesn't. And you can see they've got a lot more fat deposited in their tongue. And we wonder whether there are genes that particularly determine not only how fat you are, but where the fat is deposited. And we think there's maybe a particular distribution of fat. Now, these sleep problems I talked about, insufficient sleep, obstructive sleep apnea, shift work, sleep disorders, insomnia, are common in the workplace. And what impact are they going to have in an industrial corporate world? Because what inadequate sleep leads to, it leads to decreased productivity, it leads to increased errors, it leads to increased healthcare costs, as data showing that. If you treat sleep apnea, you see this decrease in healthcare costs. And in safety-sensitive organizations, it's going to impair safety. So there's lots of reasons that the corporate world should be interested in this, just from the point of view of their own bottom line, because they're going to get increased productivity, decreased errors, and increased safety. And how are they going to address it? Well, they can address it at the individual level, detecting people, for example, with sleep apnea, at the group level, for example, changing big education programs. At the system level, changing sleep shift work schedules so you minimize the impact. So there's lots of different ways that they can address it. Now, I just want to show you some examples here, and these are anecdotes, but I think they do make the point about the impact on transportation. I'm going to show you three anecdotes from the last couple of years in the United States that show the impact both of sleep apnea, insufficient sleep, and diurnal time. The first one is this one, the Metro North rail crash. So this was a driver of a train, and I think it was a Sunday morning, who went round that curve at 82 miles an hour. Now that curve was a 30-mile-an-hour maximal curve. And he said he was stupefied. The train derailed, there was 4 people killed, 82 injured. What happened subsequent to that is the driver was found to have severe obstructive sleep apnea, undiagnosed. And the NTSB, who investigated the crash, that's what they put it down to. They said it was to do with sleep apnea of the driver. Not surprisingly, Metro North have now issued a request for screening all the drivers. They're going to start screening all the drivers for obstructive sleep apnea, 6,500 drivers they have. The same is true in Japan. There's only been one crash, I believe, in the bullet train in Japan, and it was again due to the driver having a severe sleep apnea. They screen all their drivers. So what you're starting to see in the transportation world, Walmart does it in the US, JB Hunt does it, Snyder Trucks, the big trucking companies are starting to screen their drivers. We actually have a program with a company called Amerigas, and we're starting to screen their drivers and get them treated for obstructive sleep apnea. What Amerigas does is they've got 6,500 drivers in the US, and they're driving propane trucks. So they're driving with explosives in the back, right? So you can imagine that they are very safety conscious, because a big crash of that could cause tremendous problems. The Tracy Morgan, probably you don't know him here, but he's a famous American comedian. He's on Saturday Night Live. And what happened to Tracy Morgan is, maybe you can see here, this is a Walmart truck, and this is the limousine that Tracy was in. He was coming back from the shore. And the Walmart truck plowed into the back of him. And one of his fellow comedians was killed, and Tracy Morgan was severely injured. He's now back working. And what happened was that driver of that truck had been awake, had been driving rather for about 9 hours, 37 minutes. So he was legal. You're allowed to drive for 10 hours in the States. So he was legal. But what he had done is he commuted from somewhere down Atlanta or somewhere to get to the truck. So he'd driven to get to the truck, and he'd actually been awake for more than 24 hours when that crash occurred, right? So again, he was just insufficient sleep and not paying attention, going too fast through this area and plowed into the back of them. And here's one at O'Hare Airport. We were taking this train. It's not like the trains at Heathrow. It's a pretty basic train now. But the driver in this train fell asleep at 2:52 a.m. and she never stopped. She drove right up the escalator, right? So you're supposed to go off the platform and go up the escalator, and she just drove all the way up. There was no deaths or injury, no deaths or serious injuries. There was 32 minor injuries. But the point of showing them is one was sleep apnea, one was insufficient sleep, just real acute sleep loss, and then this was just the diurnal time, trying to perform when the clock is telling you you're supposed to be awake. Now, what we're starting to see in the US is we're starting to see big companies finally starting to care about sleep. Google, for example, you can see here, they've built these things like this where you can go into that thing and take a nap, right? So they've got opportunities for their employees to take naps. What's happened is Adriana Huffington of the Huffington Post, and she's a household name in the US. I don't know how much you know about her here, but she developed this online, publication, the Huffington Post. And while she was developing that publication, she was working all the hours. She was only sleeping 4 hours a day. She was really trying to make that thing work. And she, at some point she fell. And she fell and cracked her jaw and got fractures in her face. And there was nothing else wrong with it. And they put it down to sleep deprivation. She was just so sleep deprived. So she's now become an absolute advocate in the US for sleep. And she's written this book, which is part you can get it on Amazon, called The Sleep Revolution. And she argues that the culture change that we need is a culture just like stopping smoking, that will really begin to respect sleep. It's not a joke. We'll do something about it. And that's what she argues. And she's got all sorts of anecdotes about CEOs of other companies making terrible mistakes just because they were so sleep deprived. Aetna actually gave an incentive program for their employees to sleep. So you're starting to see things happen in terms of big companies caring about it. And I don't know what it's going to be like over here. So what I've tried to do in this talk is I've tried to say that sleep is this is a real public health problem. Short sleep is a common problem in our society. There are many, many common sleep disorders. Short sleep and sleep disorders is a problem in our workplace. There's a distinct biology to this. There's a very interesting biology. And education about sleep circadian biology is lacking, and it requires approaches at multiple levels. So thank you very much.
Thank you very much indeed, Allan, for that superb overview of the field. And it really is food for thought, isn't it? That we've missed out the importance of sleep when we're considering nutrition and exercise, and it really has its time, I think. Allan's willing to answer a few questions if people have them. Yes, please.
Well, no, there are some very interesting aspects to menopause, as you know. So one thing is, the hot flashes, for example, when you're in the perimenopausal period, they can really disrupt sleep. The second thing is that sleep apnea gets a lot more common postmenopausally. Sleep apnea is very uncommon premenopausal women. And there's data out there that looked at, this is before hormone replacement therapy was found not to do well for you when people were using it. And what was shown is the prevalence of sleep apnea in postmenopausal women on hormone replacement therapy was very similar to premenopause, and if you weren't using it, it was much higher. So clearly, women are protected, and it's even true once they get beyond menopause that the males are still, it's a bigger risk, but the menopause really changes the risk for women in terms of sleep apnea. Well, I think, obesity is the biggest single risk factor, clearly. But part of it is probably the distribution of fat. you know, I think that when you go through menopause, there's a different distribution of fat, and that may play an important role in it.
Yes.
Yes, I was wondering to informative for the public opinion, would it be possible to compare the effects of alcohol use with the effects of sleepiness on car accidents? Because if you could compare that, and if that
Mm-hmm.
that's closely compared,
Yeah.
then you make a strong case out of that.
Yeah, no. I mean, let me just say two things about it. The first thing is that there are studies out there, well, three things. There are studies out there that show that, you know, if you do direct comparisons with lapses and other performance tests, when you get to 20 hours, you're about 0.08 in the BAC, right? So you can line up the performance decrements with sleep loss, and you can, 20 hours and above, and you can talk about blood alcohol. The second thing is that the studies show that there's a huge interaction. In other words, you can take people and you can look at performance, a low level of alcohol, and they're pretty good, not that bad. A low level of sleep deprivation, not that bad. You put the two of them together, and it's got a dramatic interaction. It really magnifies the effect significantly. So what that means, of course, is that as the BAC limit drops, the role of sleep loss becomes more important, right? Because it really can amplify that small amount of alcohol. One of the challenges we've got in this world is what led to the drunk driving and all that was the fact they could measure it on the scene.
Yeah.
That they could go up, you could blow a thing, your BAC, you're over the limit, and so on. And the police are very reluctant. I mean, I've been to various meetings. Last year there was a 2-day workshop in Washington about drowsy drive and what should we do about it and so on. And the police there said they're very reluctant to charge people on the basis of falling asleep, because they go to court and they say, well, how do you know? So that we need a test. I mean, one of the things in our field is we're trying to develop biomarkers and come up with some sort of test. If we had that, I think it would make a major difference. It turns out, sorry.
In the lab, or you can set up an experiment and you can sleep deprive people.
Oh, sure.
And then you ask them particular tests.
Sure. Sure.
And then maybe you could approach it in more
Yeah, well, yeah. I mean, that's something you can do, and you're right. I mean, you can manipulate people and really look at performance decrements. The other thing is that the car manufacturers, I don't know about Britain, but the German cars, they say it's about 24% of cars sold in Germany have these systems in them that can actually detect drowsy driving, apparently quite reasonably. Because what happens when you're drowsy, and when you're normally driving, you're like this and making fine movements of the steering wheel. Then when you start lapsing, you don't do it, and you come to and you make a sudden jerk, and they can detect that jerk in action, and they know that you're actually driving drowsy. Well, the question is, what do you do with the information? And the NHTSA people in the States have done studies where they looked at drowsy driving with that technique, and people sleep deprived going around the track, and that occurred before they drove off the road on this track. So it was predictive. So there's a lot of effort going into how do you detect it, but then once you've detected it, what you're going to do about it. And the police basically believe unless we get a test that we can use the same model as drunk driving, it's going to be hard to change this.
I think we've got time for just one more quick
Sorry.
question.
I was wondering about the solution. So obviously we should all sleep 8 hours per night or something, but what about these power naps or these multiphasic sleeps?
No, that's an interesting question. I mean, so there are, the answer about power naps is that you can take these relatively short naps, right? And you can get some restoration. It's not going to be the same as you get across the night, but you can certainly improve performance. And the reason you can do that is because the recovery process is non-linear. Right, so for 20 minutes, even though it's only 20 minutes, you get this increase. And David, my colleague at Penn, David Dingy, is probably one of the better studies in napping. And it was in long-haul pilots going from the West Coast to Japan. And what they did is they wired them all up actually to do EEGs and all that, and the cockpit. And one group was allowed to nap in the middle of the flight, and the other group wasn't. And the group who weren't napping, when they were landing in Tokyo, they were lapsing and they were struggling to stay awake. So that showed that this mid-flight nap could actually be helpful in terms of boosting performance. And I believe that they actually, that's something they do now in these long-haul flights. So that's the reason. It's just you get this non-linear thing. It doesn't get you completely there, but it certainly helps.
Thank you very much
Okay.
once again, Alan.
Sure.
Okay. Yeah. Just stay there a second. I'd like to invite Professor Russell Foster, who's the director of the Sleep and Neuroscience Institute at the University of Oxford, to come and say a couple of words and to make the presentation to Professor Pack. Russell.
Thanks, Carl. We're just leaving.
Yeah.
Okay.
I don't have my glasses on. I don't have my glasses on. Could you put it on?
So you get to a certain age, right?
I sleep okay, but I can't see properly.
Okay.
Well, as Colin said, I'm Russell Foster. I'm the director of the Sleep and Circadian Neuroscience Institute. And the strategic aims of this institute, which is based in Oxford but also has connections nationally and internationally, is to understand the fundamental nature of sleep and circadian rhythms, how these systems work, what happens when these systems fall apart across the health spectrum, from blindness to mental illness, to try and develop therapeutic interventions to try and stabilize sleep and circadian rhythm disruption. And then our fourth aim is to try and disseminate this information to the broader community, both the academic and the non-academic. Now, we're new guys on the block. We're young pups. And any success that we have is because we will be standing on the shoulders of giants. And so what we felt we should do is acknowledge that contribution of individuals whose shoulders we're standing upon. And so we instituted the Sleep and Circadian Neuroscience Medal, which is for outstanding and sustained contribution to the field of sleep and circadian rhythms. And it is my immense honor that Alan agreed to accept this medal and to come here today and give us this superb talk.
Okay.
And I'll just show you the... your name is actually on the medal, Alan, with a date.
That's great.
And there we go.
Well, that's beautiful.
This is for you.
Well,
Thank you very much.
Well, thank you very much. Thanks a lot. Thank you. Okay, Carl. Okay, thank you very much. Well, thank you very much.
Thank you, everyone, for your attendance. Safe home.